Okay, we're 2 minutes in. So for those of you who have joined episode 5, this Fireside Chat from in silico to in vivo with Mike Palovich, thank you all for carving out some time in what I'm sure is a very busy day and week for many people here. Really excited about today's conversation. So first and foremost, my name is Naheed Kurji, co-founder, president, and CEO of Cyclica. Today's conversation is not about myself. I'm moderating, and so we're going to shine the spotlight on my colleague. But before doing that, I just want to read a land testament that is very important for us. We acknowledge the land we are meeting on is the traditional territory of many nations, including the Mississauga of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat people, and is now home to many diverse First Nations, Inuit, and Métis. We also acknowledge that Toronto is covered by Treaty 13 with the Mississaugas of the Credit. So with that, today, I'm really excited to introduce folks to Mike Palovich, a dear friend, a seasoned drug hunter, and somebody that I've gotten to know over the past 4+ years. I won't do Mike service or justice in introducing himself, so I'll pass it over to him momentarily, but I'd like everybody to know how excited Cyclica was in announcing a couple of weeks ago that Mike joined us in the capacity of Chief Science Officer and Head of Drug Discovery, and is now working with an incredible team of Cyclicans in the drug discovery unit and across the institution in our platforms team, our innovation team, our strategic partnership team, in helping guide Cyclica's future and achieving our vision of building, scaling the most robust and sustainable pipeline of drug discovery assets in the pharma industry. And that scale, that impact is really driven by a lot of the technology that we've built over the past number of years that Mike will be talking about today and kind of comparing and contrasting. And that's why we call ourselves a neo-biotech company, a new way of doing biotechnology, a new way of doing drug discovery. And so with that, I'll stop sharing my screen. Mike, my first question to you is, who are you? What are you all about? And tell the world your story of what brought you to Cyclica.
Yeah, well, thanks. Thanks, Naheed. And first off, I'd just like to say how excited I am and honored to be joining Cyclica and feel very privileged to be taking on the Chief Scientific Officer and Head of Drug Discovery role. So super exciting stuff. I guess, who am I? Well, I mean, I'll just start the focus mostly on career stuff. So I've been doing some kind of form of small molecule drug discovery for just a little over 24 years now. Up to this point, all of it at an organization called GlaxoSmithKline, pharmaceutical company. I often tell people that, when I first started working at GSK, maybe 6 months in, that it was like an aha moment. I was like, oh my gosh, this is for me. I love doing small molecule drug design, love doing medicinal chemistry. And I've been fortunate to continue to do it through those 24 years. I guess to cut to the chase about why I decided to join Cyclica and make the move, make the jump from GSK to Cyclica. I've been a believer in about 5-6 years now that the drug discovery industry is evolving and will continue to evolve, needs to evolve to meet demands of patients, investors, et cetera, of society in general. And I'm going to say 5-6 years ago, I started really getting interested, and I'll be the first to admit it, I am nowhere near an expert in machine learning and deep learning, but I have slowly and gradually educated myself and trained myself up, and owe a lot of favors to others for helping getting me there. That machine learning and deep learning techniques are going to play a big role, a critical role in that evolution of small molecule drug design and drug discovery. And had known Naheed for quite a while, 4 or 5 years, something like that. And when Naheed approached me and said that they were ready to take Cyclica and build a drug discovery organization and asked me my interest level, I was like, count me in, I'm all in. So that's the short end of or short story of, I guess, my career and how I came to be where I am now.
Awesome, Mike. Thanks for that background. And you come to Cyclica with a unique perspective, that being pre the world of what we now call artificial intelligence in drug discovery and development. And now you're seeing a lot of the impact and the opportunity. So let's do a little bit of a time continuum story.
Okay.
Talk to us a little bit about the key learnings that you've had over your career in the way in which drug discovery has been done. Walk us back to 1998 when you started your career, and tell us a little bit about the journeys that you went through to bring drugs to the clinic and to patients, and what the world would have looked like if you had some of the technologies today. I'd say that would be like topic number one that you can go deep on. And then topic number two is related to that. As you see the advancements in many techniques, both computational and experimental, there's so much innovation happening. What are you most excited about, and where do you think the industry is going?
Okay, so I'll try to start with the first question, which is kind of like looking back and talking a bit about some of my experiences and themes, and then trying to tie in, I guess, your point around, where were opportunities that machine learning, deep learning, computational technologies could have played a role. So hopefully I get all of it. If not, just jump in and tell me where I missed, or where I can elaborate forward. So I guess where I'll start off is that, as I mentioned, when I first started doing small molecule drug design, I told people it was kind of like my aha moment in life. I was like, oh my gosh, this is for me. And I love doing small molecule drug discovery and every aspect of it. And then I gradually learned and got more experience, more knowledge, was able to apply things forward. So to get to your question, I think the way I'll answer it is maybe pick up on some things that I've learned and experiences that I had, and then how those kind of impacted me. And I'll try to tie in some of the machine learning and in silico stuff along the way. I guess what I'll start off with is data, right? So when I think of small molecule drug design and medicinal chemistry, I think of it as almost solving a puzzle, right? And data, and spotting patterns in data. And in some cases, being able to spot the pattern is very straightforward. So say maybe you're trying to improve a physical property, you have a certain set of molecules, certain leverage of activity data or physical property data, and you could find patterns in that pretty straightforward. So my first point would be is that I learned that the ability to spot patterns in data using, I guess, is the least amount of data or the most effective use of data allowed me to make as much strides and big impacts as possible. Not only myself, but members of the team and all the partners that are involved in the team. So that would be my first response, is the ability to look at patterns in data. And then if you think about it, there are instances where those patterns and data become very complex, or the data sets become very large, very complex themselves. So at some stage, I found that my abilities were very limited in being able to spot patterns of data, or maybe even very inefficient in spotting patterns of data. And then that's where I'll tie in the opportunity around computational techniques, machine learning, deep learning, is then using those to augment my limitations in being able to spot patterns of data, and then help me do a better job at designing molecules, making decisions, stuff like that. So I think the first phrase I'll throw in there is data, spotting patterns of data, and effectively leveraging techniques. The other thing I would say is, with the evolution aspect, is I'll be the first one to admit that early on in my career, probably didn't use that much machine learning, deep learning, computational science technologies. And I would say one of the learnings and evolutions I had in my career was accepting the fact that these were valuable, learning to use them, and then accepting the fact on how they fit into my ability to then do and execute drug design. So I guess what I was saying is initially, it felt like, okay, I could do this on my own. Maybe the problems weren't complex that I was trying to solve. But then over my career, I've learned to adapt, incorporate, and appreciate more the impacts and the opportunities, to the point where I would say in my career right now, I almost feel like it's a necessity that if we're going to evolve as a drug discovery industry, that these techniques and technologies need to be accepted by a lot of individuals, teams, organizations, and then obviously, they will grow and advance even from what we have right now. So I'll stop there. I think that got the first part of your question. I have to admit, I forgot the second part of the question after my sometimes endless discussion topics, but you might want to repeat it.
As it usually goes with you and I, ask a question, and we can just continue to go down and talk about something. So the second question was, let's talk about the future. What are you excited about? But before going down that path, let's just double click on some of the learnings that you've had. So you talk a lot about data. Data is a tricky issue, right? Is more better? Is better quality and less better? At what point do we think about like our one's data strategy? And there's been so much effort in our industry on understanding data. Where does it live? How to harmonize it? How to gain access to it? How to harmonize it? How to apply it correctly? And there's a number of pre-competitive consortia who have come together with big pharma companies, with biotech and academia. Where are we on, in your view, on the continuum of data? What is the big opportunity that we now have from a data standpoint to have demonstrable impact in the way in which medicines are discovered? Is it in target identification and looking at kind of patient populations and taking all this omics data to identify new targets and biomarkers? Is it in drug design, hit identification? Where do you see the lowest hanging fruit based on your over 2 decades of experience, and then doubling down on that data point?
Yeah, so another great question. I'll probably pick and choose the parts that I feel like I'm most comfortable and experienced in answering, but I think you probably provided part of the answer already. This is when you look at the drug discovery, I guess, chevron or paradigm from, target selection. So identifying a target that people have high confidence in that is going to be, if you drug it, is going to give you the desired outcome in patients, all the way through to, drug development and getting something onto the market. There's data sets and opportunities for data all across there, right? I'll probably stay away a lot from the target ID aspect because I'll be, again, the first to admit, not my area of expertise, but my understanding and experience have been, lots of investment in the industry and genetics, genomics, and data sets, and using then machine learning, deep learning techniques, and a variety of other techniques to then try to pick out the best target or sets of targets for a given disease. And in my experiences, I've seen volumes and volumes of data come forward. And I guess one of the things I would say is that, there's a lot there, and deconvoluting it and then picking and choosing the right bets on targets moving forward is still a challenge for industry, even though we've got a lot of data coming through from that end. I'll probably skip the other end of the chevron as well, which is the development end. I have some experience in the development end, and obviously, but not obviously enough to probably comment so much on data sets. But again, the same type of thing. I've read papers and have been aware of opportunities where people are trying to learn, design the best clinical trials based on previous outcomes, data sets, you name it, to then minimize patients, try to affect the ability to design a clinical trial with the right outcomes and the right measures so that we could effectively move medicines forward through development. I'll probably then spend most of the time, probably talked too much already, about the middle part. So as I mentioned earlier, spotting patterns in data is critical. One of the things I've learned is that there's ample opportunity of data sets, both external or publicly available, plus internal, around, I would say, large areas of physical properties, right? Where people have looked a lot of molecules, across a lot of assays, where you see there's high throughputness of those assays, and therefore you could collect a lot of data. And perhaps there's enough data already out there to build models, predictive engines, whatever you want to call them, to help predict and then even design new molecules. Then I would say, as you march along from early lead discovery, getting closer to clinical selection of a candidate, your data sets become smaller and maybe even less precise or lower quality. So I'm thinking of data sets around animal PK, data sets around human PK, safety assessment data sets as well, right? There's clearly data sets out there, but pulling them together, helping them get aligned so that you could build the best and greatest algorithms to help predict or design, I would say, were the areas that I have a lot of interest in and seeing how they grow. So again, I guess to summarize, I said early on, my opinion would be, and again, it's just my opinion, lots of data out there to help you get the individual physical properties and maybe even, individual, I would say, single components of drug design. But when you get to more complex systems that we need to analyze and read out, mostly in vivo systems, that's where the data sets either become fewer, data is more sparse, measured in a bunch of different ways. You have to do a lot of energy to try to pull them together. And yeah, it makes it difficult then to apply advanced in silico technologies to help you build and design better molecules.
So let's focus in on that middle part where you are a subject matter expert or where you have more familiarity and comfort. And let's look at specific disease areas. So for example, neurodegenerative disease, I recently read that there's like 143 failed clinical trials for Alzheimer's in a row, something like that, whereby molecules that go through preclinical and animal model studies just don't necessarily recapitulate when they're in humans. So when we look at the preclinical, the amount of data, what diseases do you believe have better data that is actually now more relevant for human clinical trials? And where do you think for specific diseases where there's just not enough, or there may be a lot, but that a lot may not be of the quality that we need?
Wow, great question. Probably one I can't answer very well, but I'll tell you what my beliefs are, right? So again, focusing in small molecule drug design, preclinical small molecule drug design, our challenge is to find a molecule and then build the information, the data set that enables people to believe that that molecule will work in patients. So the actual disease system, right? So I think the way I'll try to answer your question as best as possible with, again, not focusing in any disease areas, because I think maybe each of the disease areas have varying ups and downs with regards to the amount of data that allows you to make that preclinical to clinical linkage. I think that's where the challenge is, right? Which is we need to figure out systems that allow us to predict or measure how our molecules are going to work in preclinical systems. And then that gives us confidence and value that then say we could springboard into disease systems. The other thing I'll add on to that answer is not everything's the same, right? So, and I'll just try to be very polar in this example. In some cases, you could do preclinical to clinical linkage where you have strong faith that it's a single mechanism, single target that's going to have a large impact in disease, or you believe to have a large impact in disease. And let's face it, I don't think there's many of those examples out there, but there are examples out there. And then maybe to get to where you're going is then there's the other end of the spectrum where there's very complex diseases or disease systems where it's unlikely that a single target or even a couple targets are driving the majority of the disease pathway or the disease issue, in which case then you're probably going to need molecules that are affecting more than one target, areas of pathways, stuff like that. Again, those areas now become much more complex to do in a drug discovery situation versus a project where you're looking at a single target, a molecule engaging with that, and then the direct impact. So I would say the degree of difficulty goes from maybe here to way up higher when you're going after disease areas where it's very complex and there's probably multiple mechanisms involved, and perhaps the industry doesn't even fully understand those. So it's probably the best answer I could give you. Probably not a very fair answer, and I might have dodged the bullet a bit too. So.
No, I think that at least that scratched the itch that I have on my mind. There's dozens of people probably listening to this, so hopefully it answered theirs. If folks want me to double click on any specific question, please put in the chat and I'll make sure I ask Mike, whether it be on something that we've discussed or something that's on your mind. I want to touch on attitudes and behaviors for a second. Okay. And bear with me as I kind of go down a little bit of a rant, but I think this is a fairly balanced perspective, is that at least my observation has been the market right now is kind of bimodal. You have a bunch of people who are really excited by what's going on and investing a lot of capital and time and knowledge to push this innovation forward. You have a different group of individuals who are a little bit more passe, or I'm going to use the term curmudgeons, about applying application or the application of artificial intelligence in drug discovery, and saying too much glamour, too much hype, too much sizzle, not enough steak. As a med chemist, a drug discovery scientist who has seen the befores and the after, I'm coming back and I'm kind of tying it together. This is not the first wave. While it's in the front of our minds now, the application of computational techniques to drug discovery is more prominent now than it ever really has been. this goes back 30, 40 years to some of the prominent work in computational biophysics and molecular modeling and docking approaches to drug discovery. And big claims were made in the late 80s and early 90s. There have been some fruits of that labor. And now there's a second big claim that's happening, which is today. What's the big difference between the attitudes that have existed in the 90s, the attitudes that have existed today, and what's changing? What are you seeing? I have some perspective, but I'd love to get your perspective on that.
Yeah, sure. So again, a pretty meaty topic. So changing, I'm not sure if I would use the word changing too much, but maybe I'll go with this answer and see how you respond to it. And again, I only speak to myself as a scientist, right? And hopefully this resonates with other scientists out there, and some generalizations I made won't be offending to people, right? I think scientists in general have been trained to be convinced based on data, right? Show me the data, but then don't show me once, show me 2 times, show me 3 times, show me a dozen times. And then people become more and more convinced, right? So I think in our upbringing, our training, we've been show me the data. And I think that's the same type of concept for a lot of principles, but I'll play it out to the use of AI ML to your question. I think people are like, I want to believe in it, or not going to believe in it, or I'm on the fence. But as you show me data, show me experiences where it's worked, show me instances where it's not worked, which I also think is a big thing, right? We've got to be honest with ourselves and say, here's where things where it fell short of our expectations, and here not on in addition to things where it went above our expectations, and then continually applying it, continually learning from your successes and your failures, and then continually evolving the technology is the way it's going to happen. And I think that's the way it's been, from when I started drug discovery 24 years ago, probably when I'm long gone and retired from drug discovery, is that there will continually be evolutions. There will continually be people that need to be convinced, some less more than others. But it's not different than anything else with regards to drug discovery. And for a variety of other platforms we have, where people are going to want to see the evidence, and they're going to want to see the evidence multiple times before they say, yep, I'm a believer. And oh, by the way, I'm just going to routinely use this as part of my daily practice in drug design. So it's probably the way I answer your question. Not sure if that's where you wanted me to go or were you expecting?
No,
So.
I think all your answers are the way in which they ought to go, which is, this is about you and your perspectives. So I think that's totally fair. I would add, there are 3 big themes that at least I've noticed over the past few years that have, and again, I entered the space 9 years ago, right? So I don't have the 90s, the early 2000s, and the amount of work that has been put forward prior to really entering the space. So take what I say with a grain of salt, but the conversations that I've had on understanding the history of computational drug discovery, what's changed now largely is the availability of data that we've talked about. There's a higher propensity to relying on data as a guiding principle, not just as a backstop. So data as the decision-making framework that scientists ought to use to then point to a specific direction, not just as, I believe it should be there, does the data support this? Instead, what's the data saying? Let's go evaluate that. I'd say that's number 1. Number 2 is cloud computing, right? 2012, 2013, 2014, Amazon, Google, Microsoft, and their respective clouds started to allow us to parse through this data much faster, more cost effectively, that allowed entrance of early stage biotech companies to start moving faster. What any biotech company has as its competitive differentiation to a multinational is just the speed at which they can make decisions. Within a day, we can make 5 decisions on the evolution of a company. That just can't happen. It allows us to then answer different questions. And then the third is, I'd say, education. You go now to, I have the privilege of speaking at Northeastern University and Brown and University of Toronto and Berkeley and a number of institutions, and what they're thinking about in their biophysics and their biochemistry and their med chemistry classes is the computational end of this. What is it? They have a stream of AI and machine learning for drug discovery. And so you're getting a wealth of entrants who are thinking about the world just a little bit differently. I think the combination of all 3 of those things is now allowing these types of techniques to become more at the front of one's mind instead of the back. So I think at least
totally
that's my
agree.
perspective. Sorry.
I totally agree.
I have a question online.
Oh, okay.
And I'm going to mention from who, because I have a lot of time for Gopal, Graymore, Nova Nordisk, a friend of mine, somebody I've gotten to know over the past 3 to 4 years since we met in London at an advanced AI ML conference. So Gopal, thank you for joining today. Question is specifically for Mike, considering low specificity / off targets of small molecules, we can talk a lot about that about Cyclica. Do you think AI-based drug discovery in preclinical domain is difficult to directly map into clinical development? That's question 1 of 2 questions that Gopal has asked. So Mike, I'll turn that to you.
Yeah, so I'm going to replay the question just to make sure I kind of understand it. Do you think that the AI-based drug discovery in preclinical domain is difficult to directly map into clinical development with regards to, I guess, low specificity, off-target evaluation of small molecules? It's a great question. So I'll answer it this way, right? Again, I think one of the complexities that we have in small molecule drug design is you get to the stage where you need to put a molecule into safety assessment studies, right? Animal safety assessment studies. And oftentimes you're not sure what to expect, right? And you could get hit with a result that then just squashes that molecule forward, right? So then when you walk backwards, what have organizations and what have our strategies been, is we've tried to do as many studies as possible ahead of time to try to convince ourselves or to give us a high confidence level that our molecule will be safe in those animal safety studies, and then eventually safe in people, obviously. So to get to your point about low specificity off targets of small molecules, I kind of go to promiscuity, right? So the amount of evidence that you could gather that shows that your molecule hits the target that you're intending to hit and doesn't hit a bunch of other things, so its promiscuity index is very low, then gives you confidence somewhat, I would say, that you're likely to have a better chance in safety studies versus a molecule that's just hitting tons and tons of targets, because then you've kind of, let's say, lost control, right? Now that molecule is doing all kinds of things. You've got to assume the probability is a lot of them could be bad, and then therefore you've lost control. So I guess I would say where AI pops in to this is techniques and technologies where you could look at molecules as early as possible, maybe based on structure, maybe based on physical data that you have, which then says, what's the promiscuity of my molecule across the platform of targets or platform of other indications, whatever, or not indications, but other properties that you want to look at, and then equate that to a confidence index, let's say, of having a clean safety assessment study. So I do think there's opportunities there, and Cyclica has shown some of those with its ability to look at a molecule and then say, if we look across the whole proteome, here's kind of an index of how many things it hits. Then quantifying that and relating that to a way forward, I think, is then critical. So I would say, yes, the opportunity is there, and it's definitely a tool in the arsenal.
And just to build off that, with some numbers, a given molecule when placed into a complex biological system like a cell, animals, or humans is interacting with upwards of 300 targets. And so our view, and this is not a Cyclica conversation, but the question related to something that at least scientifically we are very passionate about and consider ourselves pioneers in the world of computational polypharmacology, is our belief is that the next incremental innovation on a target-centric virtual screening drug design approach is only ever going to take a program so far. Like, what about everything else? And when we ask the question, why do oncology drug candidates fail? Well, erroneous or incomplete target ID is one of the reasons. We know that 97% of drug interaction pairs that are tested in clinical trials in oncology never advance to receive FDA approval. And in a seminal paper that came out in 2019 October, I remember reading that the efficacy of each drug that was tested in that specific work was unaffected by the loss of its putative target, indicating that the compounds kill cells via off-target effects, not necessarily their on targets. And so when we think about off targets and clinical outcomes, you need to think about a much more holistic view. You need to think about polypharmacology. That's often the common mechanism of action, especially for complex diseases like some cancers, many neurodegenerative diseases. A couple more questions, Mike, that came through.
Okay.
Your views on AI and biologics design. How does it compare to small molecules? And then from there, I have one more question and we'll get back to our conversation.
Yeah, I might have to punt on the AI and biologics designs, just, not my area, small molecule guy by training and experience. So if it's okay, maybe I'll punt on the biologics design question. We could go to another one.
Yeah, and I have nothing to add to that besides 90% of funding in AI goes towards small molecules, largely because I think just the availability of data, molecular modeling techniques have largely been used for small molecule drug design. And as a result, there's just a wealth of data, degrees of freedom make it difficult for docking-based approaches for biologics. But you're seeing an increasing number of companies emerge, many in Toronto, which I'll wave the Canadian flag to, that are looking at protein design and other larger molecules beyond small molecules. But again, scientifically, like Mike, will punt on that one. Question from Ravi Ram. Ravi, thank you so much for the question. Don't know one another, but pleasure to have you on this call. What are the bottlenecks, challenges in terms of 1, data, 2, methods for higher productivity of AI drug discovery development?
Yeah, so the bottlenecks, challenges in terms of data, and then methods for higher productivity AI. So I touched a bit on it earlier. I think, again, it's the volume of data that you can create and the diversity of your data set that's critical. And again, when I think of drug discovery, in the early phases of drug discovery, I think there's great opportunity for volume, quality, and diversity of your data set, mainly because the physical data that you could gather, the assays are fairly high throughput, low cost, you name it, right? So you could, if it doesn't already exist, or you need to augment your data set, relatively quickly and relatively cheaply build up a data set, a diverse data set of molecules, let's say, where then you could then use that to build either a better algorithm, a better predictive tool, whatever you want to do, right? So I would say in the early phases, there's really not that many bottlenecks and challenges. Maybe the challenges is to make sure that the things are done in the same way, consistency, make sure that you understand what you're trying to gather as far as the data and it's there. But then as I mentioned earlier, as you progress forward, the data sets could become much more complex. So for example, PK studies, and I could tell you, without going into specifics, some experiences, when we've looked at PK data sets, there are a lot of variables in there, the formulation, doses, types of animals, types of species, you name it. Your data set becomes very complicated very quickly, not to mention then if you're taking that same data set and another data set, and maybe they've just done experiments slightly differently, and therefore that has an impact on your data. So as you progress along, I would say again, into like in vivo type opportunities, those data sets, the challenges of those data sets now come with all those things that I just mentioned. And then therefore, even though you may have a large set of data, let's say 40,000 plus whatever you're talking about, making sure that everything was done in a similar experimental method so that then the physical data is correct or lines up correctly or is at least measured the same way becomes a big challenge, and then a big effort and a lot of activity there. So I'll stop there with regards to challenges in data sets. I guess the other thing I would add is that there's been ways where people have been, or examples where people have tried to pull data from various sources, cross-consortium type efforts where various companies are willing to plow in their data to build a better data set to help predict much more complex and difficult drug discovery problems, like in the in vivo sense that I talk about. But I still think we have a long way to go in that space. Maybe that's why there hasn't been the latest and greatest predictive models in those particular spaces, because of those challenges with those data sets. But with there's challenges, there's opportunity, right? So let's try it. Let's get there.
That's right. Thanks for that, Mike. And I know I was planning to get back to a few questions that were on my mind, but one question came through by Manjeet Singh. Hi, Manjeet. Thank you so much for joining Cyclica's M2M. And it's related to something that we talked about 5 or 10 minutes ago about neurodegenerative disease, preclinical, clinical. There's a question, I'm going to read it as it came through, and there's kind of a statement embedded in this question. So
okay.
My question is regarding the application of AI for neurodegenerative diseases. In real world scenario in AD, Alzheimer's disease, and PD, Parkinson's disease, patients have more than half of the neuron are dead by the time patients are diagnosed. Sorry, a little bit of grammar there. Half the neurons are dead by the time patients are diagnosed. It is not a matter of target, but lack of functional cells. Any take on this? There is a disconnect between preclinical models and real patients.
Yeah, so I would say that I'm not sure if you have a question there. I think you hit on a lot of the the issues or the challenges. I shouldn't say issues, but challenges of that. I think you're right. In the neurodegenerative field, again, I've have some experience in there, and I wouldn't class myself as an expert by any stretch of the imagination. I think you kind of hit the problem right on the head. Is that in these diseases where there is progression of disease over time, there's an inflection point, I guess is what you're getting into in your comment and your question, where there's almost like a point of no return, right? Or a point where you can't fight back or you can't correct the problem. And how good are we as a society in identifying the opportunity to recognize that there is the beginnings of that disease and then the window of opportunity with a therapy before it crosses over kind of like that threshold where the therapy just may not have any effectiveness or has limited effectiveness. So I guess my answer to your question, just agree with you, is it's a big challenge. It's a big opportunity in that particular space that the industry is going to have to tackle and come up with some solutions for. And I'm not saying that the industry already hasn't done so and tried many times, but I think with any of those progressive diseases, like you just talked about, not only neurodegenerative diseases, that is a common problem.
Mike, thank you, and hope, Manjit, that answered your question. I'm sure there's so much more we could go into, but in the interest of time, feel free to reach out if there's any more perspective that you want to share in a space that Cyclica is double, tripling, quadrupling down in diseases of the brain. And so thought, perspective, expertise like that, always happy to have conversations. There's a lot of questions coming through both on the LinkedIn channel, YouTube, as well as directly Zoom. I don't know if I'll be able to get to all of them, but yes, Seer, Debasis, see your questions coming through. I'm just going to take Mike down a different trajectory and hopefully touch on some of your questions momentarily. Mike, the question that I have for you is, what is success? And how do we help the industry kind of measure success? The question comes from just some perspective, and you've been sitting in on a number of partnership and prospective investor calls and starting to tell Cyclica's story and vision. And one of the questions that we frequently are asked is, where's your phase 2 asset? Where's your phase 2B asset? Have you brought something to patients that is approved in the marketplace? If so, that is how we will define success of your organization using these new techniques. Is that really the stick by which we should be measuring success as an industry? Or should we be thinking about it, in my view, and more practically? First question. Question number 2 is, within the data space, low data targets, high data targets, success will be defined differently. Take it away.
Yeah, sure. So a couple questions. So I'll try to tackle the first part. How do we define success? So again, I'll go back to one of my original statements, and I won't try to belabor it too much there, is that drug discovery obviously is a paradigm of activities that eventually leads, hopefully, to a marketed drug asset. And you just pointed out the most obvious measure of success is, can you get drugs to the market, to people, right? So clearly on that. But at every step of the way, there's a question or an unknown that we don't have that needs answering, right? So there's an opportunity for success around tool identification, because somewhere out there, somebody needs, let's say, a molecule. Now, there's obviously other ways to also validate targets, but somebody really wants a molecule, either an in vitro or in vivo tool molecule, that helps them validate a biological target for a given disease. An organization that could deliver those targets, people that could deliver those molecules, huge value there, because without that, then you might not have the further opportunity to deliver drugs. So that's one, right? Carrying on down the road, and I'll just hit another one and try to tie in your high data and low data targets, right? One of my experiences have been is that of the onslaught of new targets coming in from the genetics and genomics initiatives, there is a large proportion that fall into what I'll call the low data space, meaning that there's no protein structure, there's no known ligands, and how do we find starting points for those molecules? So let's classify those as low data, dark proteome, whatever people want to call those, right? Physical-based screening systems are in place, and sure, that's an opportunity, right? And we should absolutely do those because you could win in that space. But a lot of times you're not going to win in that space or using those tools and techniques. So what are the latest and greatest things that we could do to try to find chemical starting points for low data targets? The 3D protein prediction structure stuff is really exciting to me. And again, like any technology, it starts out at a certain level of effectiveness, it comes better and better and better and better, and then at some point it hits its this is a really great tool and technology. We're somewhere in that paradigm right there. But I would say that's an opportunity. And organizations that could use that information will then be able to have a decent to great shot, better shot, whatever term you want to use, for finding small molecule starting points for those low data targets. Again, those aren't going to be drugs right away, but they may then be enough where people then have advanced lead series for those low data targets, and then a certain organization may say more value on those than others, and then there's an opportunity to partner, an opportunity to develop further, whatever you want to do. So I would say that is another opportunity there, is applying technologies to find chemical starting points for low data targets. And then there's everything in between. So polypharmacology, a lot of things we've talked about a lot. Drug repurposing, another area. I love fast follower projects where you may have identified a particular new mechanism through your drug repurposing, but now you want to find a new chemical entity because either the one isn't practical anymore, or you want to find your own with novel IP, fast follower approaches. So there's a value there as well. So I would say if you look across the various steps of the drug discovery chevron, there's an opportunity to add value to the whole picture of drug discovery rather than just getting to the clinic and eventually to the market, which is the end goal. But in between, there's opportunities to deliver to help do the baby steps along the way. I can't remember what your second question was. Sorry.
No, I think that was the question. And if we go back to like low data targets and link it to the exciting space of protein degradation, molecular glue, there's the work that we did with the SGC on co-crystallizing decaf-1, that opens up a whole world of going after low data targets that are related to really interesting, multiple therapeutic areas and approaching it with a new modality. And it comes to Devas's question online. One more question. PROTACs, or protein degradation assays, seem to be as good as the CRISPR-Cas9, but why this area doesn't get enough attention? Can you talk a little bit about your perspective on that question?
So I'm reading the question right now. If I could get another question, could you comment on AI on drug repurposing, and if AI ML, am I reading the right one, or is there a different one?
No, that's a question from the Zoom. That's from Ravi. We can talk about drug repurposing. This one came, I believe, via our LinkedIn channel.
Oh, okay. You better give it to me again then. Sorry.
Yeah. So a question came from Debasis. PROTAC, or protein degradation assays, seem to be as good as the CRISPR-Cas9, but why this area doesn't get enough attention?
Oh, that's a really great question. So I'll just give you my perspective on it. When you look at the protein degradation space, and let's say E3 ligases and that protein family in general, let's say, I think it gets back to the point I mentioned earlier, where there may be many other opportunities for E3 ligases to be effective mechanisms, is that the right word, for advancing drug discovery? But again, people don't know because there isn't exactly a molecule that connects to one end of the ligase and then connects to the other end of your target molecule. So again, if you could find molecules that then target those E3 ligases, then people could then study those opportunities further with regards to, is there another E3 ligase or another degradation pathway molecule that we could use, a biological target and a molecule associated with it, that we don't know about right now, and that would then be the next future or the next great thing with regards to drugging other diseases, or drugging diseases where they've been difficult to drug because we haven't been able to touch that pathway yet, the biological pathway. So again, I would say my answer would get back to that target validation, tool molecule, and then eventually really well-developed or degradator glue molecule that you want to use in your particular studies and anything moving forward. There's just not enough shots on goal, I would say, right now, and there's a lot of open space that then people could go after. So I would say that would be my viewpoint on maybe why, if the right word is to use, it's not been advanced as much as potentially could be done.
And from what I understand, in the protein degradation space, much of the chemistry is done empirically through traditional experimental medchem. Have you seen many computational techniques trying to solve the problem of protein degradation?
What do you mean by computational techniques as far as biology, or as fine in molecules, chemistry?
On the chemistry side.
I mean, again, some of the stuff that I'm aware of, maybe I can't talk about. So I would say the answer is somewhat yes, but as far as computational biology, computational chemistry, I guess is what you're talking about, probably don't have greatest answers I could give on that one. Maybe I'll just answer it this way. Probably always more that could be done. We'll leave it at that.
Okay, so let's shift to the question that you had looked at, came through by Ravi. If you could could you comment on AI on drug repurposing, something that you touched on moments ago? If AI ML has come a long way, why are we not seeing much success here?
Well, again, I think it'll go back to some of the original themes that I've talked about is that, in any given time, there's a breakthrough in a particular area, right? And the initial breakthrough might be an N of 1. And then the organization or an industry says, well, show me twice, 3 times, 10 times. And then it becomes to really grow out, right? So I would say that maybe that's a potential answer to your question of why isn't AI ML used more in drug repurposing? Because maybe, we still need more shots on goal, or we need more shots that go into the net rather than around the net. As far as drug repurposing in general, I really like the idea and the strategy for 2 cents. One is the obvious one, whereas can you find a molecule that's already in the clinic and then it could be not quickly, but then used for other diseases. So obviously you get more opportunity out of one asset than maybe just going after one target. The other thing is then a lot of times it's the deconvolution of the biology, which then could lead to a better molecule because now you understand what's going on with the biology and the pathways. And then that's where I throw in the thing around fast follower approaches, because now you know kind of how the molecule works for real rather than maybe what originally tended. Or if you do the repurposing, you now see that it engages with different biological targets and mechanisms than originally appreciated. And then you could use that information to say, what if we can make a better molecule? And then you do a fast follower effort, and a fast follower effort I define as something that has like phase 2 data, which you now know your path forward, you got your goalposts set up, and now you can maybe rapidly progress using all the latest and greatest in silico AI ML technologies you can to rapidly then close the gap from the clinical asset, say phase 2, to a starting point or an early target phase drug discovery program. So hopefully that answered the question for the person who asked. If not, again, I'll be happy to follow up on LinkedIn or anything else. But I love talking about that repurposing and fast follower approaches. Definitely going to be a big component of our strategy here.
Yeah, I think repurposing also, one of the big issues around repurposing is not the scientific question, it's the commercial question on IP. And where's the IP lie, method of use, composition of matter, and then how can a company, generally a younger company, justify the capital allocation, and where does that come from, right? And we can talk about the economics of pharma in a different conversation, but really that's the question that one bumps into is, what about intellectual property? I think increasingly we're seeing some incredible companies come out in the repurposing space and trying to solve this question, as Mike just alluded to. Repurposing is one of the key strategies at Cyclica. And here's why. Drug repurposing oftentimes, typically, works through a target-centric or a disease-centric approach. And many of the AI companies that are out in the market conduct repurposing using this approach by mining the literature and omics data. More rare, infrequent are drug-centric approaches where the underappreciated polypharmacology can drive the use of a medicine in a new disease indication. And historically, drug-centric repurposing couldn't be done because of the lack of reliable methods to uncover polypharmacology. And this is something that we have spent a lot of time thinking about over the past 8-9 years in the development of our drug target interaction proteome-wide capability matchmaker. And we have a number of projects, one in which specifically, we aim to use the predicted polypharmacology of clinically tested molecules to repurpose them in a more drug-centric way. We have a couple programs that are in our pipeline right now, one for rare disease that we're really excited about, and we hope to enter the clinic over the next few months. But this is all around kind of the repurposing of existing assets to think about reimagining new use cases for those, getting them into the right patient populations faster than otherwise possible, and starting to solve disease issues that patients are desperately waiting to solve. And then the second one is using repurposing as an angle to understand mechanism by which we can then kind of recapitulate the activity of that repurposed asset, but designing new molecules that can go into a fast follower approach. So I think, we're seeing a lot of activity, progress follows, and I'm confident in what at least Cyclica and a number of other companies are doing in the space of repurposing. Okay, I love this question that came through. Phenotypic screening is a technology that has been around for many, many years, and AstraZeneca and Pfizer and GSK and Novartis have published a lot about the trials and tribulations of phenotypic screening. Question comes through, in your opinion, which strategy is best, using AI along with phenotype-based screening or targeted drug development?
Say the question again. Which strategy? Sorry, I didn't.
Which strategy, using AI along with phenotype-based screening or more targeted drug discovery and development? So general target drug discovery.
I'm going to cheat. No, I'm not going to pick either one because I think both would be my answer. But then I would classify that, machine learning, deep learning for both, not just phenotypic, but for also target-based. We already spent a lot of time talking about target-based. Maybe I'll just spend a little bit of extra time around the phenotypic space. I mean, drug discovery is difficult enough when you're just trying to identify a molecule that binds to a single target and not everything else. It becomes, multiple fold harder if you're working with a phenotypic assay where you don't know what the target is, or your belief is that there's multiple things going on. Again, my answer will go back to data. In those systems, you generate data and you build a diverse data set that then allows you to then pick out the patterns, that then allows you to identify the molecule that works best in that phenotypic system. Now your gut check comes that phenotypic system then also translates to what happens in the disease in people, right? So that's still the gut check, but building that data set, building that smart, diverse chemical data set around that phenotypic assay, and then leveraging computational techniques, machine learning, deep learning, to help you then spot the patterns and design the best molecule is something that I'm very interested in. And I think, there's opportunity there. Why the industry kind of moved away to single target? Smarter people than me, wiser people than me probably could answer that question. Will the industry come back? My bet would be yes, is that there'll be some doubling back to saying these phenotypic systems may be more predictive of what's happening in disease. But again, it's that leap of faith of going from a phenotypic-based in vitro system to a disease system is still a big gap.
Totally. And one of the big challenges coming out of phenotypic screening, not only the translation of the phenotypic assay to human disease, is what's the target by which, the target of action, the mechanism of action. Can we explain that? Can we deconvolute that to support lead optimization or other activities? And, we've run into a number of conversations where people have really solid data. They understand their molecule, they understand its activity, but to get through regulatory Q&A and get, IND enable and get that into the clinic, the question then becomes, what is the target of action? And I remember reading a presentation, a very long, comprehensive presentation by a senior scientist at Pfizer a number of years ago, who gave a case study on 3 molecules. One in which it took Pfizer about 15 years to deconvolute the mechanism of action of a molecule coming out of a phenotypic assay, where they were really confident about its activity, but just they could not answer the question by what target is it actually having this activity? 6 years for another one, and about 12 months for another one. And so really that time frame spans, and therefore could add a lot of time. So target deconvolution that could then lead into new chemical series. Lead optimization, I think, is also another big opportunity for companies that have phenotypic assays as the basis of their technology. Okay, so we have 4 more minutes, and Mike, I want to leave you with this one question.
Okay.
And sorry to Carlos, you asked a question. Don't have the time to answer it yet, but it's a good point on drug repurposing. Feel free to ping on LinkedIn, and we can take this one offline. Mm-hmm. There's a lot of students that listen to these M2Ms. And for those of you who have been with Cyclica for the past 3 years, before it was Molecule to Medicine, it was AI Ate My Homework. AI Ate My Homework was the first franchise of trying to inspire a wave of thinking for high school students and early in their career university students and postdocs. And we started to see a big demand about what we were talking about in the application of AI and drug discovery and development. So I'd like to leave this with you. Inspire the next generation. What is the piece of advice that you have for students who are asking their question, what should I do? I'm interested in this intersection. I don't necessarily know where to apply. Where should I go?
Yeah, so inspire. So I'll do my best, right? So a career in drug discovery is wonderfully fulfilling. Trust me. Of course, wonderfully fulfilling in the sense that if you happen to be fortunate enough to get a molecule onto the market and you were part of that team, obviously wonderful, fulfilling there. But then there's so much in between where the fulfillment is great and makes such a great career. There's so many little baby steps where there's success and thrills and learning and being part of finding a solution to a problem, to a disease problem, you name it. So that would be my first thing. It's a wonderful, great career and a lot of fulfillment. On the flip side of that, it's also very frustrating, right? You will fail a lot. So I would tell people, be prepared and be comfortable with failure. The key is, though, also to learn from your failures. So if you're okay and able to say, yep, I'm going to fail a lot, but I'm also going to learn and I won't make those mistakes again, or we'll learn from those failures, keep that in mind as well. The other thing I would say is that the reason why I have loved my career in GSK is people. It takes a massive amount of people, a great team to be successful in drug discovery organization. So when you embark on your career, if you decide to embark on your career, you will have the opportunity to build up your abilities and engagements with people, learn to interact with people, grow and develop along with colleagues and others that are in your teams. And those life experiences will take you a long, long way and make you a better person in the end round as well. The last thing I would say is in the drug discovery field, there are always problems to solve. So if you're up for challenges, there's going to always be a problem to solve in drug discovery. There's always going to be a problem that we need to overcome, a better solution, something that's better than what we currently have, or something that's completely new. I firmly believe that 5 years from now, drug discovery will be much different than what it is right now. 10 years from now, who knows what it'll look like, right? So think of it that way, is that if you're joining early on in your career right now, you have the opportunity to change the way drug discovery will be done 10 years from now, and you'll be part of that. So we've got 1 minute left. I'll leave those responses there. Hopefully that was inspiring enough for people.
And I love that answer. And all I would add is if you're entering the space of drug discovery and development, take some data science courses. You don't need to be a computational X, Y, and Z, but I think empathy is important. This is very complex. Computational folks need to understand the life sciences. They need to have empathy towards the complexities of biology and chemistry, vice versa, the opportunities. If you're strong in biology and chemistry, what can compute do to support the way in which you do discovery? So it's 1 p.m. Eastern time. Really appreciate everybody joining. There's one correction that I want to make just for the record, given that this is live, is when we were talking about that drug repurposing program, and I was giving that example at Cyclica, I said a few months, a few quarters. I just want to correct so that there's no mismanaged expectations. I was speaking fast to get the point through, but really excited about the work that we're doing there. So Mike, thank you. You're a gem as always. And for anybody who's interested in learning more, go to our website, ping us on LinkedIn. Very friendly crew at Cyclica. Happy to answer any more questions. So thank you all. Cheers.
Bye-bye, everyone.